Good morning, everybody. First of all, I would like to thank the RTMFM webinar organizers for giving me the opportunity to talk about dynamic nanoscopies and how we use them in the team to shed the light on virus assembly in host living cell. As you all know, viruses are small objects made of several hundreds to several thousands of molecules, which sizes range between 20 to 500 nanometer for the biggest one, also called giant viruses. Because of the small size, they are difficult to be visualized by classical photonic microscopy. Therefore, researchers have been used for a long time now electron microscopy in order to resolve their structure and to identify them. I've represented here several of these viruses electron micrograph, such as HIV, influenza virus, the now famous SARS-CoV-2 virus, which is responsible for the COVID-19 worldwide pandemic, and the Ebola virus. In the team, we are mainly interested in the HIV virus, and all that I'm going to present afterwards is linked to this virus. HIV is an envelope virus, which is made of a core surrounding the nucleic acids, or the genetic material, and a corona of proteins lying just beneath the membrane, the lipid membrane. This lipid membrane derives from the host cell, and in these lipid membranes are embedded some envelope or so-called envelope proteins, which are the one used by the virus to target and to enter new viruses. As an example, if you want to make images of this envelope protein at the surface of the virus, so HIV virus is approximately 120 nanometer diameter. If you want to make an image of these envelope proteins using confocal light microscopy, this is the kind of picture you will get in the end. So clearly, due to the lack of spatial resolution linked to the diffraction limit, you will never be able to observe individual proteins. If you are now able to turn to super resolution microscopy such as STED or STORM microscopy, then you will start seeing individual objects on the surface of the virus. Nevertheless, although you gain clearly into resolution, you're still very far from what you can see using electron microscopy, when there you can clearly see at the same time these individual envelopes, the lipid membrane envelope, also the core of the virus, which contain the genetic material. In the team, we are mainly interested in the assembly of this virus at the surface of the host cell, in that case, living T cells. And this assembly is a dynamic process. And although electron microscopy gives access to very thin and interesting details of the structure, it doesn't give any information about the dynamics of the system. So assembly mainly consists on the ability of each gag molecules that are the structural proteins of the HIV virus to be able to find other gag molecules in order to self-assemble, starting from few of them up to 2,000 to make a new virus. This has an apparent size ranging from 2 nanometer for the single protein up to 120 nanometer diameter for the fully assembled virus. And the time needed to make a new and to release a new virus is known to be approximately 30 minutes. To study this process, we decided to use multiple particle tracking approach coupled to palm microscopy. In this simulation, where particles are moving, you can see that using this multiple particle tracking, you can achieve many trajectories that will give you information about the dynamic behavior of each single molecule, but you can also have information about the average position of the molecule over the whole time. And this by projecting all the position of the molecules over the whole acquisition time. It shows you that you obtain some density enriched, which are significant of poorly moving particles. We applied this approach to HIV gag in T cells, and we track under palm microscopy each individual gag T cells inside the T lymphocyte, ending up in thousands of trajectories and some density maps on the surface where we could identify individual ongoing assemblies. We analyzed this approach by gathering the time and resampling the time of each acquisition. So we took one image every 20 milliseconds to be sure to capture all the diffusion events of each single gag, but then we resample by gathering the 10 first seconds, and we reconstruct a movie of all the trajectory 10 seconds after 10 seconds during the whole time that will cover usual assembly time, which is from 10 to 20 minutes. By projecting all the position of the particles, we could identify some localization accumulation. While performing this is on the left, the density of the position over the whole time of the acquisition, and these are the tracks. We started to observe some striking and interesting tracks that seem to be directed towards the high density position. The issue with that kind of behavior is that it is very difficult when you perform tracking, whatever is the algorithm you use, even if you use the famous and very good multiple tracking trajectories software from the Marguet team in Marseille, it is still difficult to know if these connections are linked to the density, to the local density, or if they really describe the trajectory of one single particle. For that reason, we went to Jean-Baptiste Masson and Mohamed El-Beiri, who previously developed a new method based on hidden Markov states and Bayesian inferences called the Inference Map, which gives you the ability to analyze the trajectory based on the probability they have to be the same at the step n plus 1 compared to the step n. So we performed that approach, and we divided the trajectory into two types of motion using the Langevin equation to describe the motion. So after graph matching to recognize the trajectories, we perform Bayesian inferences, and then we tessellate the map of the localization in order to end up in areas where the average number of trajectories are equivalent one to each other. This is inherent to the Bayesian method to be correctly performed. Based on this Voronoi tessellation, we then applied the Langevin motion, and we ended up into two different types of maps. One map that described the potential local potential that could be responsible for the directed trajectories we have seen previously, and the other one, which is the diffusion map, which is without directionality and without an attractivity. By monitoring the changes in this attraction energy during the time course of assembly in living cells, we try to decipher which was the contribution of each component involved in the assembly. Interestingly, we observed that a mutant of gag-gag interaction considerably decreased this attraction energy as compared to the wild type, while at the same time, the addition of the viral RNA into the cell did not change significantly this attraction energy, meaning that the self-assembly can occur independently of the presence or not of the viral assembly. Nevertheless, by comparing the time where the density is maximum to the time where the attraction energy is maximal, we could observe a coordination process of this viral RNA compared to the other assembly where the viral RNA is not present. We took benefit of these dynamic nanoscopy methods to tackle another questions about the role of plasma membrane during HIV assembly. GAG, the main protein of assembly, is known to bind pi45p2 lipid in the inner leaflet of the plasma membrane, while envelope has been described to partition into rafts that are domains enriched in sphingomyelin standing at the outer leaflet of the plasma membrane. So the question is how these envelope and ongoing assembly of GAG are able to retrieve themselves, and two models has been proposed. One is that GAG use pre-existing lipid domains to self-assemble, which is spontaneously partitioned into raft-enriched sphingomyelin and cholesterol domains. And the other one is that GAG is able to generate his own lipid domains that will be able to trap pre-existing outer leaflet lipid domains that could be enriched into envelope. To tackle this question, we use another approach of monitoring the dynamic of molecule, which is the fluorescence fluctuation spectroscopy. So by looking at the change of intensity into areas and self-correlating the fluctuation of this intensity with time, you are able to have an idea of the average time of presence of the molecules within the area you are sampling. If this average time is short, it means that the molecule diffuses faster, and on the opposite, if it's long, then the molecule diffuses slowly. This approach, which is a local approach, has been transposed to a kind of a mapping approach by performing scanning FCS, where you can simultaneously observe different spots and monitor changes, local changes at different positions of the diffusion. And this finally has been turned to state microscopy in order to decrease the area you're sampling and to be able to access to local changes in very small area, which turns to be very useful in the case of HIV. So in our case, we perform scanning state FCS over the ongoing assembling virus, and we monitor the change in the different lipids that are involved into either the RAF domains or the binding of GAG to the plasma membrane. So we look at sphingomyelin, pi45p2, cholesterol, and we also use PE as a control. We perform this kind of approach with either infected cells that contains GAG, the envelope, and the viral RNA, or transfected only cells that contain GAG, the viral RNA, but not the envelope. Interestingly, we could observe either on infected cells or on transfected cells a strong decrease of the diffusion of PIP2 when the PIP2 is located into the area of the assembly of the virus. This appeared to be the same for the cholesterol, where we observe from 2 to 3 times differences into the median diffusion coefficients. But on the opposite, we could not observe any changes neither on sphingomyelin nor on phosphatidylethanolamine, which is a control lipid, not known to have any interaction with the virus. With that kind of approach, not only you can have access to the diffusion coefficient, but also you have a distribution of all these diffusion coefficients at the different position, either at the position where the assembly occurs or outside the assembly. And interestingly, using this approach, you can decompose the different diffusion observed, the diffusion coefficient observed, into 3 major families that are exhibiting very slow diffusion coefficient, lying in between 10 to the minus 3 to 10 to the minus 2 micrometers squared per second, that could be considered almost solid, which that they belong to strongly trapped molecules into the assembly. Or you have an intermediate state, or you have a fast state which is more related to non-trap molecules into the assembly. Interestingly, if you look at the average number by plotting the change in the cumulative frequency when the virus inside the assembly site, compared to outside the assembly site, then you can have a kind of a confinement index that you can compare directly either on transfected or infected cells. And very interestingly, what you see here is that, in fact, for PI45P2, we could not observe any strong differences between the infected or the transfected cells in terms of this confinement index. On the other side, the cholesterol seems to be much more confined where the cells are infected, which is when the envelope is present. And interestingly, we saw no differences at all in the sphingomyelin, nor we saw differences in the phosphatidyl ethanolamine. So to end up my seminar, I hope I have convinced you that the dynamic nanoscopy really shed lights on the molecular processes occurring during assembly of viruses, which are object below the diffraction limit of classical photonic microscopy. So this helped to monitor the assembly processes during the time course within the host cells. To end up this webinar, I'd like to thank all the people who took part to the work I've presented. In particular, people from the team, Delphine Muriot, who is co-heading the team, Charlotte Flotterhair, who did a PhD and performed all these single molecule experiments with the support of Jean-Baptiste Masson for data analysis and software development, and Jean-Baptiste Sibareta for microscopy in Bordeaux. Peggy and Naresh, who took part also to the lipid project with the support of Christian Engeling in Oxford and Jakob Sosnaki in Oxford, who developed and performed the infected cells scanning state microscopy in Oxford. And we perform here in Montpellier the scanning state microscopy on transfected cells. All the other people from the team that are still working with us. And of course, I thank the RTMFM network and also especially the IMABio network who provide us many support in terms of a Master 2 internship. And finally, I thank you all for your attention and I'm ready to take questions.